Well, my research is not really here in the school in Korea. All my research is now based in IAMN, and more particularly, we developed 3 years ago a laboratory, a biological laboratory within this electronic institute. And it's very interesting because you can now imagine to create something in the clean room, an electronic device or a macrosystem device, etc. And you can bring it directly within the institute in the biological laboratory and to test if this device is okay or not. And if it's not okay, to go down to the clean room and rebuild a new one. And that's very interesting because you don't need to transport it from one institute to another one. Yeah. You've got directly the biological test within the Institute of Microelectronic and Nanotechnologies.
So, when did you start your research?
Oh, it's a long time ago. I don't really remember. I think it's more than 12 to 13 years ago we started that.
It's really a long time.
Yeah, and till that date, we developed and we followed four PhD students. It means that all the things that I explaining to you were kept in consideration and were developed by students, PhD students, and there were 4 of them.
What kind of machine do you need in your research?
What kind of?
Machine.
Devices.
Devices. Oh, well, as I said before, we especially developed 4 kinds of devices. The first one used electrophoretic properties to bring parasites and to transport it within a droplet. That's the first one. And within these parasites, we were able to concentrate parasites within this device. We were able to concentrate parasites to catch a part of a drop and to transport this drop with an eWOD, it's electrowetting device, and eWOD systems to transport this part of the droplet from one part from one point to another one. That's the first device. The second one used electric properties of the parasites to identify the concentration of this parasite within a droplet. That means that you've got millions of parasites, and we were able to, through an electric analysis, to determine the concentration of the parasites. That's the second device. The third one was a device to determine if a parasite is alive or dead, and more particularly, if this parasite is able to develop infectivity. That means that this device would help us to determine, for instance, if a drug is efficient to decrease infectivity of the parasite or not. And the fourth device, which is currently in development, is a device to help a little piece of mouse intestine to grow in an artificial liquid. That means that currently we were able to develop a little piece of mouse intestine. We fed this explant with a specific liquid, a nutritional liquid. And currently we are working on microfluidic systems to feed the explant slowly and on a long time without any degenerations of this device, of this explant. Okay.
What is the main application about your research?
The main application?
Yeah.
Well, at the beginning, it's especially for the genetic analysis. The main application is to determine if there is a risk or not, and the way of transmission of these parasites in France. For the development of microfluidic device or electronic device, it's especially to develop new tools to detect the parasites, to concentrate it, and as I said previously, to determine if a drug is efficient or not. Because one thing that I didn't say to you before is that no drugs are really efficient against these parasites. That means that if you are contaminated, you are not able to take something, the medicines, to cure you. One molecule was more or less used in United States, but it's not really efficient in France. It's not allowed in France. But the lack of drug is especially due to the fact that we have no way to test many drugs at the same time, in a high throughput systems. And the device we tried to develop is to do this kind of analysis automatically. That means that you will have 100 holes with cells inside, test the drugs and the parasites, and the cells said, okay, this parasite is destroyed or not, this parasite infects me or not. And we are able to identify if cells are infected through the electric fields that passing through the cells. And if the electric fields are modified, that means that the parasite is active. That means that the drug is inefficient. Okay for you?
We have saw the recent report of you have upward is a three-dimensional control of IDOT.
Yeah, and what's your question about this article?
So we'd like to know when you do this research, is there something interesting or some challenging happening?
Oh yeah, for this article talking about the three-dimension culture of crypto, the main challenge was in the fact that the explant of the intestine of the mouse, before this experimentation, you can imagine that this piece of intestine were destroyed very quickly. That means that you were not able to maintain it in its structures more than 1 week. And after that experimentation, we were able to maintain the structures more than 40 days, not 40 years, 40 days. Yeah. And that means that during that period, we were able to infect the explant and to see the development of the parasite, and more particularly, the first development of a cancer was also observed, because previous studies have shown that the parasites were able to induce a cancer in the intestine and in the colonic part of the intestine. And that means that here we've got an in vitro model to study the process, how the parasite is able to produce a cancer or to develop a cancer in the mouse intestine. And the main challenge was the time that we could maintain alive and structures this part of mouse intestine. Okay, or other questions or?